All right, pre-AP biology. We're going to continue here today with our topic of population genetics. Um, before I get into today, so I want to just kind of go back. Oh, where was it at? Here we go. I'm going to go back through just a couple of pieces of review here. The frequency of the dominant and the recessive alleles. I want us to make sure we remember, kind of what this is. So remember, if you have a certain population or a population sample, as long as you know how many dominant alleles there are in the gene pool, and you know how many total alleles there are in the gene pool, then you can figure out the frequency of the dominant allele. And you can do the same thing with the frequency of the recessive allele, right? If you know how many recessive alleles there are, you know how many total alleles there are, you can figure out what the frequency of the recessive allele is. And also remember, P plus Q always equals 1, okay? All right, so now let's get into today's stuff. Now I'm going to introduce you here to an equation at the bottom of this first slide here. I need you to write that down in your notes. I need you to learn it. You need to memorize this equation. P squared plus 2PQ plus Q squared equals 1. This is known as the Hardy-Weinberg equation. And what it basically tells us is that in a population, I don't care if it's rabbits or lizards or whales or humans, it doesn't matter. If you're studying an individual trait, like let's say fur color in rabbits, there's a certain percentage or a certain frequency of those rabbits that are going to be homozygous dominant, right? Big B, big B. There's a certain percentage that are going to be heterozygous and a certain frequency that are going to be homozygous recessive. And according to this equation, if you add those frequencies up, you will get 1 or 100% if you're dealing with the percentages. So this Hardy-Weinberg equation, what it basically is a breakdown of the population. P squared represents the individuals in the population that are homozygous dominant. 2 times P times Q represents the individuals in the population that are heterozygous, and Q squared represents the individuals in the population that are homozygous recessive. So if you know what P and Q are, if you know the frequency of the dominant and frequency of the recessive alleles, you can figure out these other components of this equation, and they should add up to 1. Let's go ahead and practice this. You'll see this. So it says the following data was taken from a population sample of rabbits in Maryville. So here we have a different population. All right, we studied rabbits in Crown Point, a couple days ago. So here's the breakdown. 35 of those rabbits are homozygous dominant. 50 of those rabbits are heterozygous, and 15 of those rabbits are homozygous recessive. So again, we can figure out all of these values here, all right, for this population size or population sample. So the first thing we can figure out is what P and Q are, right? We know that 35 of those rabbits, I probably should use B's, but I use T's here for some reason. 35 rabbits are big T, right? So they contribute 70 dominant alleles to the gene pool. 50 of those rabbits are heterozygous, so 50 dominant alleles and 50 recessive alleles. And then 15 of those rabbits are homozygous recessive. So again, they contribute 30 total recessive alleles because each one of those rabbits has 2, right? 2 little t's. So again, all we have to do is add up our dominant alleles. So we get 120, right? 70 and 50. We can add up our recessive alleles. So that comes out to 80. And then we can just figure out, again, what our P and our Q values are. All right, now when we use that Hardy-Weinberg equation, we're going to use the decimals here. Okay, we're not going to use the percentages, we're going to use the decimals. So if I want to figure out, what P squared, 2PQ, and Q squared are, all we have to do is simply use these P and Q values and just base everything off of that. So P squared, for example, 0.6 times 0.6, right? That gives us 0.36 or 36%. Q squared, 0.4 times 0.4. That would give us 0.16 or 16%. 2PQ, 2 times our P times our Q, that would give us 0.48 or 48%. So make sure that you just double check your numbers, make sure that they do add up to 1. That's always a good idea. And now when we look at this data, we can ask ourselves, what does this data tell us? All right, so again, P and Q, right? Those are the frequencies. That's the commonality of those alleles. Remember, I said if you reach your hand into a bag and pull out an allele, what's the chance it's going to be a dominant allele? In this case, it's 60%. What's the chance it's going to be a recessive allele? In this case, it's 40%. Right? Different populations have different frequencies of those dominant or recessive alleles. Remember when we did Crown Point the other day? The rabbits in Crown Point, the frequency of those alleles was different. Now our P squared, our Q squared, and our 2PQ, this is a breakdown of our population. Now remember, our population is the entire group. It's not just this sample. So what this tells us is that 36% of our rabbits in our population are homozygous dominant rabbits. 16% Q squared are homozygous recessive, and 2PQ, 48% are homozygous, or I should say are heterozygous. So again, I go back to this equation here. Oops, right here. Oh, sorry, where is it? There it is. Those percentages we can plug into this, okay? And that, if you add them up, you get 100%. All right, so again, that represents our population. In our population, we have a certain percentage of each individual genotype. That's what those numbers are representing. All right, but again, always make sure that those things add up to 1. Make sure your P and Q add up to 1. Your P squared, your 2PQ, and your Q squared all add up to 1 as well. All right, guys, that is it for the notes for today. I'll see you soon. Take care.